she's the cute one. She's the one that would snuggle with everybody, right? So Amanda, that was Amanda's favorite from the first litter. All right, sorry, I forgot to start the recording. Amino acid synthesis. Amino acid synthesis starts around our favorite pathway, right? You might recognize this as glycolysis, and you might recognize this as the Krebs cycle. So all of the amino acids spin out of these two cycles if we're going to make them from scratch, de novo. Okay? We can recycle. The recycling isn't really that complicated. Pretty much when you eat something, the acid hydrolyzes the amino acids. Amino acids very readily permeate through the gut as a nutrient, and then you can recycle them. Derivatives commonly filter into this glycolysis pathway or into some of these other pathways. So if it gets, if an amine group gets knocked off or whatever, they very readily go into these pathways and are recycled. So there's not really much of a salvage pathway like there is with nucleotides. We generate all of these, and we'll go into detail on all of them. On most of them, I guess I should say. And they very readily are just generated that way. Oops, did I go backwards? Okay. Most of this occurs in our mitochondria because that's where all the cycles are around, right? So, and we do have some that occur out in. Oh, one other quick point that I should point out. Oh, it's not on this slide. I'll point it out. Oh, actually, it's this point. Okay. We'll notice that this all happens in the mitochondria. We do have things that, there's very common pathways that branch off. So citrate comes out of the Krebs cycle here, right? And it goes into acetyl-CoA. Acetyl-CoA can go into leucine and lysine. We also can spin off and do fatty acids and steroids, as we'll note in the later lectures, from the same molecule here. So a lot of these are just backbone structures that we end up just placing functional groups onto. Isocitrate goes to alpha-ketoglutarate. Alpha-ketoglutarate, again, you might recall, is part of the citric acid cycle, but it doesn't get permeated or doesn't pass through the membrane. But isocitrate can get converted, and then alpha-ketoglutarate can go to glutamate, and from glutamate we can generate proline, arginine, and glutamine. It doesn't always make sense what you can generate amino acids from. So, for instance, so from here, this glycine, serine, cysteine. Serine is how we generate glycine, which doesn't make much sense because you would think glycine is the easiest amino acid. All it is hydrogen. You wouldn't think that you would generate. You think the reverse. You think you generate serine from glycine, but actually we generate glycine from serine. And so it's not always clear how this happens, but you'll notice that from pyruvate we can generate lysine, valine, and isoleucine, and isoleucine takes a different pathway than leucine, right? So again, not always intuitive as to where these come from, but just know the important part of this slide is two things. First off, a lot of this stuff happens in the mitochondria around the Krebs cycle, and second off is a lot of these are bare bone molecules that have multiple functions, and so they can go into fatty acids, they can go into carbohydrates, and various other things, polyphorins, steroids, all kinds of other activities. Because it's a biochemistry class, I need to talk about nitrogen fixation. Okay? Does anyone know how much percent of the air is nitrogen? It's 70% of the air is nitrogen. But what form is that in? It's a gas. It's this N2, right? And then what do we use for all of our biological functions? What form of nitrogen do we use? What's that? It's ammonia, right? We use ammonia. So we have to take it and go from nitrogen to ammonia. And there's an intermediate where we can make it nitric oxide and a few other intermediates. Okay, but there's a chemical process that occurs. It's going to take it from our nitrogen to our ammonia, and you can see it's pretty costly cycle, right? It takes to get 2 ammonia molecules out of this nitrogen. It's going to cost us 16 ATP. It's a pretty costly cycle. We don't actually do it. That's the irony of it. As humans, plants and bacteria are the ones that do nitrogen fixation, and we just eat them, and that gives us our nitrogen, right? But it's a pretty important process. Basically, there's 2 enzymes that do this. We have a reductase that has iron, and we have a nitrogenase. So we're going to take electrons from reduced ferredoxin are going to come in, and from there we're going to use a nitrogenase, and we have we're going to take the nitrogen, it's going to be converted into ammonia. And ATP does not power the reaction directly, but it's used to make the exergonic reaction kinetically feasible. So it's not using it; it's just it's not directly using it in a transfer. It's just using it as background energy. So nitrogenase complex here are the 2 enzymes that we just talked about. So this is the iron protein reductase, and here are the spots for ATP. And here we have our iron core that's held into place by cysteine residues, and this iron sulfur cluster transfers electrons from the ferredoxin to the nitrogenase. So that's how we're going to transfer electrons over here. When we transfer electrons, what kind of reaction are we talking about? It's an oxidation reduction reaction. If you see an iron molecule, that's probably a good indication that we're talking about an oxidation reduction reaction. We all know this intuitively when we look at rust, right? It's a very powerful oxidizer, and that's primarily what iron and sulfur together do a good job. So this is the molybdenum iron nitrogenase. This is a heterotetramer. There's 2 types of cluster. We have a P cluster, and then we have this molybdenum iron cofactor. Let me shut off my phone. And those 2, there's 4 of them. There's 2 clusters inside this tetramer. You can see there's a lot of iron and cysteine in these, a lot of sulfur in there, and that's held together pretty tightly. The P cluster accepts the electrons, right, from the iron reductase, and then the molybdenum iron center reduces the nitrogen to ammonia, and that's what this is. So this is homocitrate. Here's the histidine residue, just like in where we see another cysteine residue that interacts. Well, the molecule has a histidine residue that interacts with an iron cluster. Use it to breathe. Hemoglobin, yeah. Hemoglobin. It's not exactly the same concept. I mean, what molecule does the histidine in hemoglobin interact with? I'll just give you a hint of it. They love hemoglobin. Hemoglobin's structure was characterized. It was the first one. I think it was maybe myoglobin, but hemoglobin, myoglobin, whatever. It was first characterized in, and I say they, I mean ACS for your fun. So myoglobin and hemoglobin were some of the first proteins characterized, and so they love them everywhere on those tests, just so you know. So that's why I bring it up. So histidine interacts with an iron molecule, not a molybdenum molecule there, but the idea is pretty much the same. We've got a cofactor. What's the cofactor in hemoglobin? It's the first part of it's heme, right? So hemoglobin has a cofactor heme. Heme interacts with the histidine residue and an iron molecule that allows the oxygen to go on and off. So we have this molybdenum iron cofactor that's going to take the ammonium and make it into, or the nitrogen, and turn it into ammonia. Okay, so that's going to generate the nitrogen. We have scavenging molecules that we're going to pull up the nitrogen, and from there we generally are just going to use it by popping off the amine groups from amino acids. That's pretty much how we're going to, as mammals. Plants and animals can take ammonia directly, or generate that ammonia. They're going to interface it with alpha-ketoglutarate, right? Where do we see alpha-ketoglutarate? What cycle? The Krebs cycle. Yep. We can use NAD or NADPH as cofactors, our coenzymes, and a proton, and we're going to make glutamate by adding that ammonia group here, and then we reduce or oxidize this NADH, and we make water. So what kind of reaction is this if we have water here? It's a condensation reaction. And then we use glutamate as a nitrogen source for the rest of the amino acids. And you knew that because we also use it for what other cycle do we use a lot of glutamate for as a nitrogen source or to solve in catabolism? Urea cycle, right? The urea cycle uses glutamate and glutamine. So does our amino acid biosynthesis. I just answered my own question. Okay. Glutamine synthetase then can also incorporate another nitrogen into glutamate. And what are we going to do if we add another nitrogen? We're going to make glutamine, right? So we can make this glutamate can be this carboxyl group can be made into, what kind of group is that? Or we have a carbon-oxygen double bond and an amine. What kind of functional group is that? Yep. Amide, amide, right? That's what an amidation reaction is, right? Where we convert a carboxyl group to an amide. Okay? And basically all we're doing is taking off we form this intermediate. This phosphate is acyl phosphate intermediate, and then we're going to take and we're going to add in the amine group and knock off this phosphate, and it becomes a full-blown phosphate that can be used in another cycle. Glutamine is a very versatile nitrogen donor, as is glutamate. But glutamate synthetase is the enzyme that's going to facilitate this amidation reaction. This is a different way of showing that same picture, just very directly. Citric acid cycle produces oxaloacetate. Oxaloacetate produces aspartate. Aspartate, this is the nice part of this reaction, is it shows that we can make asparagine, methionine, threonine, and lysine, all from the oxaloacetate and aspartate. Threonine can be used to convert to isoleucine. Citric acid cycle can also produce alpha-ketoglutarate. From alpha-ketoglutarate, we can make glutamate, and from there, we can make glutamine, proline, and arginine. Pentose phosphate pathway, ribose 5-phosphate, we can make histidine. Glycolysis, we make phosphoenol pyruvate and erythrospore phosphate. From there, we can make phenylalanine and tyrosine. We can also directly make tyrosine, and we can make tryptophan. Glycolysis, 3-phosphoglycerate, serine, we can make cysteine and glycine. That one's every time I see that, I'm like, wow, that seems totally whacked to me. Glycolysis, pyruvate, we can make alanine, valine, and leucine. This is kind of a quick snapshot reference material for you. So these carbon skeletons from our catabolic pathways provide intermediates, and that's the glycolytic pathway and the citric acid cycle, as well as a pentose phosphate pathway, or the pentose phosphate shunt, and those provide all the intermediates that we can then use to be metabolic precursors for amino acids. We have certain amino acids that are essential, meaning we can't make them. Okay? At one point in your amino acid charts, you asked whether you need to know them. You don't really need You don't really need to know them. And I say that, I'll look at the test again and see, but I don't see much value in memorizing them. Just know that these are amino acids that we have to have in order to, as part of our diets, in order to, because we do not have pathways to generate some of these other amino acids. This is not necessarily how we make amino acids. Let's just put it that way. We don't have all of these pathways. These are how they're made, but we don't have all of those enzymes. Why would that be? Why would we not have the? It annoys the crud out of me if you want to know the truth. It seems sloppy. Why don't we need to have those enzymes to produce those amino acids? Yeah, natural selection has just kind of removed that option, or never had it in the first place. We just don't need them because plants or whatever is just going to provide those amino acids. Here are the essential amino acids, and it's kind of crazy the number of steps in the pathways of some of these. So, non-essential, we have a number of steps in the pathways here, right? So some of these take 16 steps to get through and make one amino acid. That's quite. So not just that we don't need to. Is it energetically favorable to make them make your own? I mean, you can see why, right? So the questions, some of these take 8, 9, 10, 16 steps. So essential ones, we stopped manufacturing basically because we didn't need them. And you can see that the reason why we don't need them is they're very energetically costly to do. And so we can avoid doing it, and they're already prevalent in your diets. And it's kind of some of the reason why it's important. Some of the reason why we have evolved to be omnivores. A deficiency in one essential amino acid can have severe ramifications, right? So you'll see this in specialized diets, or there's certain people who have deficiencies in these, which most famous is, we've talked about it, PKU, phenylketonuria, which is basically where the enzyme that converts phenylalanine into tyrosine does not work. I think we've even showed this slide. 16,000 people in the U.S. and Canada live with PKU. So some of your, anyone drinking a Diet Coke or anything in here? Some of your diet soft drinks have phenylalanine in them. And for people with PKU, that's a big deal, right? PKU, but if you don't have this enzyme, what happens? Okay, this is an exercise now. Your job to figure out, someone needs to tell me what this disease does. It does, but explain to me, what's the disease pathology? How does this disease, what's happening here? Look up on the Google real quick. Someone tell me what's going on. I can tell you, I just want someone to, I need a little bit of interaction from you. I'm going to ask you to step out of your comfort zone a little bit. Yes. Okay. And? How? It's just an amino acid.
It causes damage to some part of the system.
Okay. Phenylalanine builds up. Is it the fact that we don't have any tyrosine, or is it the fact that we don't have enough phenylalanine? You have too much phenylalanine. Okay? Which basically says what about molecules. We've talked about what happens when you have too much glucose. Is glucose a good or a bad thing? Both. If I get one concept through to you here, dosing is very important. Is phenylalanine good or bad for you? Both, right? Dosing is important. It can be very important. Phenylalanine, good for you in the appropriate doses, right? In a healthy person, you need some. The rest is converted over to tyrosine. If we don't have this phenylalanine, hydroxylase, it can't add on what functional group is added in the phenylalanine to tyrosine reaction? Hydroxylase. Yes, a hydroxyl group or an alcohol group, an OH. That's what's added to phenylalanine to make tyrosine, right? So it can't do that. That's what this enzyme does. And so phenylalanine accumulates, and that's what causes the damage. There's an BH4 is tetrahydrobiopterin. That's a cofactor for the phenylalanine hydroxylase. That actually, that's one of the things you can test for. Sorry, I thought I had a little slide on that. So there are inheriting PKU. You can get that from both parents, or you have to get that from both parents, because if you have two non-PKU carriers, that's not a carrier. You can have, this is standard Mendelian genetics, right? Probably learned all this, when you were a sophomore in high school, right? So not a carrier, quarter or half are a carrier, and a quarter have PKU if you have two parents that inherit a PKU gene. And there's a whole spectrum of stuff in between. Again, phenylalanine hydroxylase is going to add on this hydroxyl group onto this tyrosine, or add it onto the phenylalanine to make a tyrosine. There's a recycling pathway there. Again, healthy person is going to process excess phenylalanine through phenylalanine hydroxylase and make tyrosine. If you have a deficiency, you can't make it, and so the phenylalanine accumulates in blood and tissues and can cause neurological damage. And phenylalanine is in lots of things. Right? Fish, beans, diet soda has a ton of it because is it aspartame? Aspartame has a derivative of phenylalanine. So that's what you see on the Coke bottle cans. They'll tell you phenylaltronics or whatever the term they use for that reason. Wheat, eggs, all these things are high phenylalanine. So if you'll see people who have PKU have a very specific diet. So how we test for that, normal person will have phenylalanine from dietary proteins, so they'll have a high amount of that. They'll have, the blood levels will be safe though. A person with PKU in a normal diet would have too high levels of phenylalanine, that would be toxic. A person with a PKU in a low protein diet will have low phenylalanine from dietary protein, and they'll have low levels of phenylalanine in their blood. So that's kind of the... And my favorite part, the part that I am growing to be more and more interested in, is this genomic aspect of things, because there are 200 known mutations associated with PKU in that phenylalanine hydroxylase. That actually strikes me as a very large number. And so, even though there's not a lot of people who have this disease, only 16,000 people, the number, that's a very interesting number of mutations in that associated with PKU. And what that means really is that the penetrance is, I saw several slides that show, and I couldn't find, there's a graphic that wasn't very clear, otherwise I'd have put it in there. There's people who have more or less severe cases of PKU based on the type of mutation you see in this gene. I don't know what you're going to see as far as pharmacogenomics goes. I don't know what you're going to see in those classes. but this is a classic example of things that you're going to start to see a lot more of. We're going to start to know what, instead of saying we have side effects, we're going to start to note that certain side effects are caused by certain mutation or genotypes that exist within the genome. So certain people can, we're already starting to see this with like SSRIs. We're starting to see that certain people can process SSRIs better based on a certain enzyme pattern that they have. And so you're going to start to see this kind of analysis done quite a bit. Where people come in, I think within your lifetime, I think probably within 10 years of you practicing, you're going to start to see genomic profiles of people, and you're going to start to notice, okay, they have this genomic profile. Because they have this genomic profile, they can take this drug or they can't take this drug, or this drug is more effective, this drug has this side effect. You're going to start to see that kind of profiling, and this is just the start of it. This is just an example of how we're going to start building that up more and more and more. I think yesterday's lecture, especially that TED Talk, should give you a vision of where you're going. For me, it's very, very exciting. I get really excited when I think about that because, frankly, well, you guys have a problem. I think I have a grandson that's going to be born. We found out yesterday it's going to be a boy, which I have mixed feelings about. Because boys are idiots. I can say that because I'm a boy. I have first-hand experience of that. I'm sure some of you have first-hand experience of that. I'm sure some of you have second-hand experience of that. You don't have to justify it. I don't have to justify it. Anyway, so I found out that we're having a grandson, and I'm really convinced that within his lifetime, he will have the opportunity to live as long as he wants. You know, if something catastrophic happens to him, that would be the exception, right? But disease-wise, I think we'll be able to grow organs. I think we'll be able to identify diseases, and I think he'll be choosing his. I think my great-grandchildren will be able to be designer babies. I think that's how crazy it's going to be. I think he'll have to make that decision. He will be making those decisions. So. And this will be one example. This will be an example of a disease that just goes away. People just won't have it anymore because we'll just edit out those mutations. And yesterday we were talking about the number of genotypes that we have like 500 pages. The whole thing makes us unique. That number is going to shrink, and you're going to see certain things like, hey, I want to be able to breathe underwater, so I want to have an alligator hemoglobin, right? I want to be able to. Because alligators can breathe underwater for, I don't know what it is, like some insane amount of time, right? So I'll incorporate that ability into, like I want my kid to be a swimmer or whatever. I don't think, you guys can debate the ethics of that. I won't. But I think that will be an option for you guys. We have a tough enough time with just our stupid news media. You guys are going to be making significant decisions. Hopefully you guys do a better job than we do because we're a bunch of idiots when it comes to making decisions. So, as adults, I can speak for my entire generation when I say we are idiots and assholes, and we deserve whatever. You guys do not deserve what we do to you. And actually, my parents are even worse. The baby boomers, like they're just screwing everybody over big time. But that's a different topic for a different day. Okay, so, and they always gripe about you guys, right? They always gripe about, oh, the millennials, they're so this, and I'm like, you've got to be kidding me. You guys are. You realize how much deficit you've put into place. You guys want to acknowledge global warming, climate change. I mean, I'm just going to list of things that are like your generation. Sorry, I didn't get on a rant. Okay, amino acids from transamidation reactions. So, glutamate, we get it from our diet, right? We also can generate it through oxaloacetate conversion. Oxaloacetate plus glutamate is going to be aspartate plus alpha-ketoglutarate. Pyruvate plus glutamate is going to be alanine and alpha-ketoglutarate. So we can generate aspartate and alanine from glutamate, and our intermediates from which cycle? The Krebs cycle. Yep. These cofactors are all associated with the enzymes. So these are important vitamins that you probably will start to see, especially like vitamin B6. Those are important vitamins that are essential. Glutamate's central to all this, as I mentioned when we talked about the urea acid cycle. Glutamate comes in the form of glutamine, or can also be transferred amongst our cells as glutamine. Glutamine will be stripped of its amine group to form glutamate. From there, we can make a GABA. Some of you are probably familiar with GABA as a neurotransmitter. Did we talk about GABA? Have we talked about GABA as a neurotransmitter? Did the men talk about GABA as a neurotransmitter? Sometimes we do and sometimes we don't. It just depends on what case studies we talk about. But GABA is a very common neurotransmitter. So glutamate can also be converted by gluconeogenesis into glucose. We can make glutathione, or we can enter into the Krebs cycle here. We can also enter into the Krebs cycle at the between the Krebs cycle and the urea cycle and ornithine, and go out and process through the liver. Glutamate plays a central role in all of these processes. It's central to physiology. It's one of the ways that we move nitrogen around. It's obtained from the diet, from our diet primarily. You can see also that it's also very important as neurotransmitters between two synapses here, and it can also be transported in and out at these junctions, and they play a critical component in synapse in transmitting signals. So, and we also have glutamate receptors that are straight up glutamate receptors. What do we need glutamate for? Primarily, though? What molecule or atom have we been talking about all day long today? Good guess. The other one that's in the air. Nitrogen, right? We use it primarily for nitrogen. Hemoglobin's a good guess, but we did talk about it a lot. Okay, next amino acid. Serine. So 3-phosphoglycerate, right, that's a glycolytic intermediate. It's a precursor for serine. So we take 3-phosphoglycerate. Is this oxidize or reduce? Is it gaining electrons or losing electrons? Gaining, so that's reduction, right? So we're reducing NAD plus to NADH. We end up with 3-phosphohydroxypyruvate. Okay, there we're going to add glutamate. That's going to be, we're going to remove the amine group. That's why we're talking about glutamate first, is because glutamate and glutamine provide the nitrogen or the amine group for most of the amino acids. From there, what enzyme would be most likely going to be moving the nitrogen from the glutamate, the amine from the glutamate onto this 3-phosphocserine? What enzyme? Talked about last week, so I know that was an attorney to go, and I'm putting you on the spot. What enzyme moves? Good guess, no? Transaminase, right? So I'm giving you most of it. It's an amine, so it's a transaminase, right? Okay, so it moves. We're talking about moving nitrogen from glutamate to something else. The transaminase, right? The transaminase is going to move the amine from glutamate onto 3-phosphocserine. 3-phosphocserine is going to be involved in a hydrolysis reaction, and we're going to end up with serine. So that's how we end up with serine. Now, again, and serine is important because it's required for cysteine and glycine. Tetrahydrofolate is an important cofactor in this process. It's derived from folic acid. What is folic acid important for? Yes. So when we're making a lot of stuff, tetrahydrofolate, folic acid deficiency, folic acid is one of the primary vitamins that you give to pregnant women, right? From there, we could generate glycine, right? So the serine hydroxymethyltransferase, and here's the cofactor, the important cofactor tetrahydrofolate. We're going to take serine and make glycine. It basically is going to transfer off that hydroxyl group, off that glycine. We can also make from threonine. We can make glycine. We can also make glycine from glyoxylate, and we can also process it in the glycine cleavage system down to bare bones, which is carbon dioxide and ammonia. So these are all important, or all pathways that we can make for creating and degrading glycine. Again, serine is really important. Its intermediate is 3-phosphoglycerate, but it can be serine's used for. It's a very important amino acid for general proteins. We can also generate glucose and glycine, alanine, cysteine, cysteine, choline, ethanol. I'm going to go through these. What are all these? Sphingomyelins, phosphotidylserine, selenocysteine, ethanolamine, choline. What are most of these? Let's try. No. Proteins, right here. It's okay. I need you to guess. I need people to guess at least. Try to process what's going on. So I encourage you to take a guess. They are having to do with membranes. What composes membranes? Spent an entire week on it. Lipids and fatty, and more precisely, fatty acids, right? So, and what kind? Storage or structural? Structural. Structural are used for what? Membranes, right? Storage are used for. Yes. For energy, right? Right? Which is the second half of this lecture. Okay. We also have 1-carbon metabolism that's associated with this. 1-carbon is methylene formyl, right? This is a carbon double bond to the O, and then just a hydrogen for the other bond. Formula amino, right, which you have I think it's we don't use this one very often, but I think it's a double bond, that carbon. Does that make sense? Nope, that'd be a single bond. No, double bond to the nitrogen has the hydrogen. So that's what that's formamino, and that's a group that gets transferred around, and then methylene, where you have a double bond here. So obviously we have our oxidation states are most reduced all the way up through our most oxidized. So we commonly see these, we'll see these methyl and formyl groups. That amino I'm curious, obviously amino, what's the amino acid amongst the group? Which amino acid is really an amino acid? You can guess. Good guess. Which one of the amino acids is an amino acid, not an amino acid, which the rest are? There's 20 amino acids. One of them is very different than the others. Which one? It's an amino acid.
What are you saying? I don't know.
It's okay. I'm all right. An amino acid versus an amino acid.
Oh, okay. I had no idea what you were
It's fine. That's why you are welcome to ask me anytime.
Proline?
Yes, proline. Proline is our amino acid, and I don't know. I've never seen a functional group that's what that would be, right? I've never seen a functional group that's associated with that. In fact, this is the only time I ever see that. But I suspect there may be some kind of enzymatic process that uses that as a transferring entity. But so part of this process, one carbon metabolism, or we don't have a lot of one carbon metabolism, just so you know, right? We don't have a lot of that. Normally we take them off in pairs, but we do have some. Tetrahydrofolate is amongst that process, and we basically convert it using tetrahydrofolate, and we've got this very complex. So we have tetrahydrofolate here, right? And it can go in between N5, N10 methyl tetrahydrofolate. Right? We add in this one carbon here. That one carbon can be converted down here into a double bond, and from there we can make N-formal tetrahydrofolate. We can also go down through this pathway. I don't know the value, and I'm just showing you this as a tetrahydrofolate because tetrahydrofolate will be something that you will see. You're going to have doctors will prescribe these kind of the folic acid for pregnant women. This is the pathway, and this is one of the processes by which one carbon, I'm not going to test you on it, and you will not see that in the final. It's just kind of a frame of reference for you. This is not something that I dive deeply on a lot. SAM, which is S-adenosyl-methionine, is the major donor for methyl groups, and S-adenosyl-methionine is synthesized from methionine and ATP. And here's the reaction. So basically we have methionine, right, and ATP, and we're going to make this. What is this right here? This blue structure right here. Yes, it is. And so we have this methionine attached to this nucleotide, and that's the major intermediate. And that's what does it do? It helps transfer it's a major donor of methyl groups. So this methyl group right here is where you get it's a donor for. So here's an example. So we have SAM. Right, we're going to donate this. Here's our one carbon that gets a methane group that gets cleaved off into whatever molecule. From there we end up with adenosyl homocysteine, and from there we cleave off in a condensation reaction or a hydrolysis reaction. We take adenosine, right, and we make homocysteine. Homocysteine is an intermediate for several other amino acids that we're going to talk about here in a little bit. So methionine is regenerated from homocysteine and SAM, and that's part of the methyl cycle, which we'll talk about here in a minute more. Actually, right now. So that allows us to transfer these methyl groups around. So adenosyl, methionine, we have basically a cycle that's going to occur between these four, and we can spin off methyl groups from those. And we can transfer those methyl groups in to make all kinds of molecules, especially amino acids that use that methyl group. Again, back to the pregnancy thing, we have the methionine cycle, where we're going to methylate certain molecules. Those are going to be used to generate proteins. We have the folate cycle. Here's what we were talking about earlier. We also have RNA and DNA synthesis that also use these purines, use some of the tetrahydrofolate, as you might recall from yesterday. And then homocysteine sits in here, and it also, homocysteine is used as a endoplasmic reticulum stress indicator. Indicates DNA damage. Homocysteine in your blood is not at a high level is not a good thing. It can also be used as a transsulfuration molecule for serine and shouldn't say for our serine. Homocysteine can be used with serine to generate cysteine that can be used in proteins. So these pathways are very, very important. So there's a lot of medications that interfere with the folate metabolism, and so there's a lot of reasons why you would take those. Like in particular folic acid, because some of these medications interfere with those. So that you will see that, in fact, some of the drugs that we've talked about earlier, like metformin, I think we talked about fluorouracil and sulfasalazine. Some of these other ones that we've talked about are used in birth. I don't think we've talked about birth control pills interfering with folic acid. That's another reason why it's really important that you have a balanced diet full of folic acid. So these things definitely impact the folate cycle. And folate cycle, again, really important not only in homocysteine directly impacts this folate cycle, as I just noted also there's tetrahydrofolate is also used in other cycles, but it's also used in DNA and RNA synthesis. So it's also used in a lot of different places. Obviously, there can be a lot of impacts with that. As such, homocysteine levels are a great marker for problems. So if you have high homocysteine levels, you're at greater risk for cardiovascular disease. It indicates that there's a lack. This is very relevant for you guys because this will be an indication that you have side effects from some of your drugs. So a lack of cystathionine synthase activity, that's an indicator of that, and that's the enzyme that converts homocysteine into cysteine, which we show here with the serine. So it's a very, very important indicator, not only just in cardiovascular disease, but also just important for a lot of metabolic activity. All right, let's take a break for a little bit. And for 5, and then we'll come back. So English.
Next Thursday.
Maybe second.
Yeah, it's coming up quick.
3 weeks.
Yeah, come back to 23rd.
I'm sure Long knows what he's doing. Does he December 7th?
Say,
Long, does he December 7th?
I don't know. I think he does it every so often. Because he went to that university, it's like Sichuan University, it's like honors. So he went there and then I think worked there for a while. So I don't know if he does it every summer or if they do the program every summer. This is the first time I've heard of it. So maybe I met like a criteria with me that I
How long have you known Long?
it's about the same time we've been working with Amanda.
Did you meet him?
Through the
Oh, really?
Yeah, she was like, yeah, you should. Because I was looking for advisors at the ME, but I decided I wanted to do it. She's like, yeah, you should work with him. So
I
okay,
like
yeah.
Long a lot. We've done several. We did the senior summit
Yeah.
together. I really liked it.
He wants to do one for like soap collection, the secret design, the collecting it from the device.
We should actually, I should get with him.
You should.
We should put another senior summit. Matt and I should put another senior summit together.
Yeah.
But
He's
maybe
a
with
good
him,
person.
maybe it's Long. I mean, it'd be a lot of fun.
Yeah,
We should do an electricity.
You should, yeah. That would be pretty cool. You can update the one that he's got. His is gigantic.
Well, we could just use the microwave device that you just add in that
No, I think that's a good.
electricity. It really wouldn't be that hard.
Yeah. Concepts there. I think you'd be able to do it. Yeah, at the he goes there. He's teaching a course this summer through that program. It's like a 2-week course on composites. But he was talking about putting Taylor as a secret designer again because I helped him with one where we were taking silk and making those heart valves shapes. That was more straight up material science, you know, real application.
Oh.
I think doing the collection would work with the electrospray.
What if it is two different teams? Do you think there's an interest in that?
I think so. I mean, it doesn't have to be specific to silk with collection. It can be fiber collection with any
You
extract
could take
process.
collagen, keratin, and cellulose hair and respin. We had a grad student do that at Utah who did that.
Someone was
He
working
was
with
at
it.
the barber, but hair every day.
Yeah. Someone was doing that when I first started at the department. It made one of the labs just explode.
Oh, it's more stuff. Yeah, it's hair is pretty.
I think it's a good idea. So I'll be permanently updated with the
Yeah.
I told Long that I wanted to help even just promote mentor some students if they need help. So I really enjoy doing it.
Yeah. See how this fall versions so that would be interesting. All right, should we get that rolling so we can get out of here? Okay. All right, so we're talking a little bit about regulation. And you will see this type of question on the final. And I'll give you an example of how this works in the practice of the study session that we do next Wednesday. Not next Wednesday. 2 weeks from Wednesday. 2 weeks from today. So if we have an enzymatic pathway, A, B, C, D, blah, 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 down to Z, commonly it's regulated by the end product. This step here is called the committed step. Okay? So once it gets past that, that's a committed step. There's no way to inhibit this reaction anymore. Does that make sense? So the inhibitor is commonly this product. It's inhibited, so it binds that enzyme. Once that kind of makes sense, right? It's kind of like I liken it to transactions. If I need to buy milk, I look in the fridge. There's milk in there. I'm not going to buy milk, but once I don't have any milk, I'm going to go buy milk, right? Kind of makes sense. That's the way this pathway works. If I produce a whole bunch of Z, I have a bunch of milk in the fridge, I'm going to inhibit going to the store and buying it. This is the committed step between A and B. That's the end step. That's the definition of a committed step. For example, we talk about serine synthesis. The committed step is the 3-phosphoglycerate dehydrogenase, which happens to be inhibited by the end product, which would be what? In serine synthetase. Serine, right? So serine inhibits that enzyme. If there's a high concentration, it's going to do that. Here's kind of that. Here's that serine. This is 3-phosphoglycerate dehydrogenase. Serine binds into the regulatory domain, right? The catalytic domain is down here. The regulatory domain is right there. That serine binds in there and inhibits the 3-phosphoglycerate dehydrogenase, which happens to be the first, some of the first steps in that pathway. Same thing, here's an example of threonine. So threonine deaminase, right? We have, that's the starting. Here's the committed step. It goes from threonine to alpha ketobutyrate. That goes down the pathway. Isoleucine can be the inhibitor. Valine can activate it. So that's because it's also part of another pathway. So it can have multiple feedback mechanisms that inhibit or activate. That committed one is usually the first one, the first step. So it can be a very complicated mechanism of inhibition as well. Since it has multiple things that can come out of it, as you can see here, we actually can make leucine, valine, and isoleucine out of this process, and this is all part of another complex process. That committed step needs to have cumulative feedback, so it needs to be regulated, but it also has to have multiple mechanisms, so like valine and isoleucine, in order to regulate it. So this kind of gives another example where you can have branched, and all of this occurs in enzymes in spades. So if we have, we need to go from A to X or A to Y. Y can inhibit enzyme 2, X can inhibit enzyme 1, and yet that multiplicity can still allow that pathway to go, but it can also have allosteric properties, it can have different committed step properties, and it can also gate the reaction so that only a certain amount is going to work and another amount is not. Okay, so that's enzyme regulation. And you'll see that in with the amino acid synthesis. All right, so now we're going to talk about fatty acid synthesis. This is where we're going to make large fatty acid molecules. So this is a cycle. Right, so we have certain entry points, acetyl-CoA, malonyl-CoA. We're going to have a cycle in here, and out of that cycle, we're going to generate large chain fatty acids. So we're going to add 6 or more times to get C16, 7 more times to get C18 from this already that already has 4. So this butyryl also can go in there. So we have multiple ways of generating the same thing, the same molecule. And I'll have a little bit better picture. This occurs in 3 stages. So there's a stage here. It's in yellow, that's the initial stage. We have this stage, and then we have our repetitive addition. So we can activate the acetyl-CoA and the malonyl-CoA in this step to get to butyryl ACP, and that allows us to get to the third stage, which is the repetitive addition. And all of this occurs on this ACP molecule, this butyryl ACP, and you can see that there is a thiocysteine there, or thioester there. That ACP is an acyl carrier. Okay. Here are the 6 the principal reactions that occur. This is one of the charts that you can just use for your reference. There's the reaction, the step, and the enzyme. I'm not going to go over it because, frankly, I don't know that it's that exciting.
You'll see it 6 times in here anyway. Here's the same reactions in a little bit different way, but the enzymes are a little bit more clearly laid out, and you can clearly, a little more see clearly how this becomes a cycle. So this cycle just keeps. So we go, here's the acetyl-CoA, malonyl-CoA. We enter this cycle, and we're going to add in 2, 3, 4, 5, 6 additions. And basically, we're going to add in 2 carbons at a time to generate these long-chain fatty acids. So in the first round, this is where we go. This is where we activate, right? First round of synthesis. This is where we go from our malonyl AC. This is where we go from that down to the butyryl ACP. We end up. I like this chart because it tells what kind of reaction we have. So we have a reduction, a dehydration, a reduction. This is the reaction sequence. We have condensation, reduction, dehydration, reduction. I would kind of know exactly what we're talking about as far as being able to identify the type of reaction. You're likely to see that. So this is the first round of synthesis. We're going to go from acetyl ACP down to butyryl ACP. This is where we activate it and put it on the scaffold. So citrate, which is synthesized in the mitochondria, is going to be punched out into the cytoplasm, and it's going to get cleaved by ATP citrate lyase, and that's going to generate acetyl-CoA for fatty acid synthesis. So we have this reaction here occurring. So this ATP citrate lyase is going to generate this acetyl-CoA, and that's going to be out in the cytoplasm. That's going to be the first step. Acetyl-CoA for fatty acid synthesis. NADPH is our reducing agent, right? It's a little bit different than NADH. We do have NADH in there, but NADPH is going to be the major reducing and oxidizing agent that's going to be used to form these molecules. Pyruvate is converted into oxoyl acetate, and from there, malate dehydrogenase and pyruvate malic enzyme and pyruvate carboxylase are going to then generate the NADPH. Here's where we start getting into this fatty acid generation. So we've kicked out, right, the citrate that goes into our acetyl-CoA. That's going to be then used in to generate our fatty acids. After that, the NADPH is going to enter the cycle here, one from the pentose phosphate pathway of the shunt, right? Or we have malate to pyruvate. Both those are going to kick in the fatty acid metabolism. So citrate exits, goes to acetyl-CoA, and this is where we're going to start. All in the cytoplasm, all of that, a lot of the products get kicked back into the mitochondrion. That allows the pathways, and we have cooperation between all these different pathways. So malonyl-CoA is synthesized by acetyl-CoA carboxylase. That requires biotin. You may have, where else have we talked about biotin? I'll give you a hint. It was in the proteins. It's protein-protein interactions. It forms a very strong reaction with what molecule? Good guess. It forms a very, very, I think it's even on the test. I think I even had this on the test question, right? It forms a very, very strong reaction, almost covalent interaction with avidin, right? Streptavidin, neutravidin, just avidin in general. So biotin-avidin is a cofactor, and this is where we're going to convert our acetyl-CoA to malonyl-CoA in order to get the activation going. Fatty acid synthase is a very complex molecule with multiple domains. It basically provides an acyl carrier, as I mentioned earlier. So this is the structure of the fungal fatty acid synthase. Here's a mammalian fatty acid synthase. You can see that there's different
functional different domains in here. There's a nice little chart that shows what the different charts do. So KR down here is the beta keto acyl reductase. So that's right here and right there. DH is the dehydrogenase. The KS is the beta keto acyl synthase. That is right there in the middle. So anyway, you can go through the whole thing. There's malonyl palmitoyl transferase. Basically, these whole reactions occur. All of this reaction occurs in one enzyme in the reaction chamber. So this whole kind of pathway thing is just, it's like crunching through all four of those reactions and doing them really quickly. And that's all happening within the fatty acid synthase. So we have a condensation reaction for the ACP, right? And then we end up with a reduction, a dehydration, and another reduction. And basically that keto group at the third carbon is converted to a methylene group, and that's going to form your butyryl ACP. So now we're done with the second step, right? So an NADPH is a source of that reduction. Second round begins with condensation of malonyl-CoA with the newly synthesized butyryl ACP, and we're going to form a C6 beta keto acyl ACP, and we end up with another reduction, dehydration reduction sequence. It's repeated, and synthesis continues until we have. So this is the second round, right? So now we're in the repetitive stage. This is the third step, right? The second round. We're adding more and more and more and more and more of these on until we add all the way up to C16, right? Until we end up with that C16 acyl ACP. So in this repetitive stage is where we're going to start. This is the second, third, fourth, fifth round. We're just going to keep adding on the same thing over and over and over to that molecule. It's this reduction, dehydration, reduction sequence is we just keep doing it over and over and over. And this is where what are we making here? This fatty acid synthesis. What are fatty acids composed of? What molecules or atoms? Is there anything special about them? What's the structure of a fatty acid? Okay, let's take a step back. What are proteins composed of? Amino acids, okay? What are fatty acids composed of? Or they're just long chain hydrocarbons, right? There's nothing special about them. They're just C. Occasionally we have what with them? It's a little different. Yeah, we might have a double bond in there, which makes it saturated or unsaturated. Unsaturated, right? So we may occasionally have a saturated bond in there, but frankly, they're a really boring molecule. And all we're doing is just, when we make these things, we're just chunking on 2 carbons at a time. It's really that boring. So phosphopanthine, this is one of the acyl carriers that's associated with as a coenzyme. They're part of the reactive units that you're going to see that allow us to do part of the reaction. So I just would know that. I would just know that there's certain cofactors and coenzymes in this, and know that the phospho. I'm not going to ask you to know the reaction. Just know that this is part of as an acyl carrier, and it's part of the reactive unit. It's an interesting side note. Does anyone know what triclosan is? Okay. I actually started using so I did a pathology postdoc, and I studied bubonic plague, Yersinia pestis. And one of the major antibiotics that we used was triclosan, this is the structure of triclosan. So this is how I know about triclosan. Triclosan is also used, and it's really easy to make. Okay, there's the structure of it. About 10 or 15 years ago, there was this big antimicrobial phase in soap, where they started putting antibiotics in soap in your hand washing thing. So your moms probably were all involved in that. I say that because, frankly, I'm a dad, and I didn't really give a rip. I was like, I'm washing my hands, maybe I want to, right? If it's antibiotic, great. If it's not, I don't really care. I just use a bar of soap for everything, including shampoo. I was like, you know, so I don't really care about that kind of stuff. My wife and daughters have all this fancy shampoos and blah 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 blah. Their hand soaps are all antimicrobial. That's the age of triclosan right there. It was dumped into everything, right? What are the consequences of that? Yes, and if we start doing that, right, what else happens to our skin and everything else? Right, we're killing everything off. That's not necessarily a good thing. So the mechanism of action also of triclosan is to bind to the enol substrate of the reductase, where we get the NADPH cofactor, and that's the mechanism of action for in this fatty acid synthesis cycle. That's the mechanism of action for that. So it's an interesting side note for all the. They don't prescribe triclosan. You can just go down and you can buy it by the truckloads, I think. So, all right. So the synthesis of palmitate. We're going to take acetyl-CoA, 7 malonyl-CoA, 14 NADPH plus, and we're going to regenerate a palmitate, which is a C what? How many carbons are in palmitate? (16), right? Plus 7 carbon dioxide, 14 oxidized NADPs, 8 CoAs, 6 molecules of water. So you can go through and adjust the stoichiometry and see how much. Basically, we're using quite a bit of ATP out of the whole reaction. Reactions of fatty acids are similar in E. coli as they are in animals. Again, here are all the domains in that function of the fatty acid synthase. The dimer, 2 identical chains, has 2 compartments. That's the dimer. There's a selecting and condensation compartment, which is going to bind your acetyl and malonyl substrates and then condense them, and the modification compartment, which carries out the reduction and dehydration activities, which is part of the elongation or the repetitive step 3 part. Here's kind of the structure, as I mentioned earlier, schematic of how all this kind of fits in. All this kind of interacts with this reaction site here. There's a condensation compartment and selection process here. I think the takeaway from this is these enzymes get to be pretty complex. I have no idea how something like this would evolve. I mean, the evolution that would be required to come up with this whole structure and function has got to be outrageous. So, one of the things you're also going to see in relation to this is tumors. They require a lot of fatty acid synthesis because the one thing that they're generating is a lot of membranes. So we can take some of our beta-keto-acyl synthase inhibitors, and that's going to help reduce the effectiveness of the cancer to replicate. So it doesn't really kill the tumor cells, but it retards the growth of them. So we've done several mouse studies with that. They did show dramatic weight loss, so we might also be able to use it for obesity. And again, they might be a good chemotherapy. Who knows? But there's definitely something into this. Beta-hydroxybutyric acid, when it's attached to ACP or CoA, is a fatty acid. That's what it's used for fatty acid synthesis and degradation. But it's also a ketone body,
and there's an isomer of this drug that's a very lethal, it's potent and illegal drug. This gamma hydroxybutyric acid. And, I'm not actually familiar with this. So I don't. Why don't we look that up real quick? I want to know what that is. I looked at this and I didn't think much of it until I just standing up here going, "Hey, what is this?" Look that up and see what that is. Okay. I don't know what it is, to be honest with you. I just looked at the slide on potent illegal drug, and when I was standing here, I was like, I wonder what that means. This is right out of the textbook, so I will look that up tonight and see what it means, a potent illegal drug. What does this look like?
I don't know,
Well, what's this group right here? It's a carboxyl group, yeah. What do we have here? So what are we missing for an amino acid? An amine. And what amino acid would that be? Serine, right? That'd be serine. So I'll look that up and see if there's what that means. Fatty acid synthase cannot generate fatty acids that are longer than C16. We have other enzymes for that. They are generally attached to the endoplasmic reticulum. We only add carbon in units of 2. So that's because this malonyl-CoA is a substrate, and that's how we take it off and add them in. Long chain. That's one of the important limitations. We also, when we metabolize fatty acids, we also do that in 2s as well. If we're going to add a double bond, here's an example. This is the sterile. If we're going to generate this oleoyl-CoA, right? There's an enzyme. If we go from stearate to oleate, we're going to introduce double bonds into that. They're going to be attached to the endoplasmic reticulum. Mammals like enzymes that introduce double bonds beyond carbon 9. When we say carbon 9 double bond, what's the notation for that? How do we notate that? The fatty acid. At position, let's say we're going to do position 9. Okay, that's one way of doing it. What is the other way of doing it? This is an omega fatty acid, right? Right? Omega-3, omega-6, omega-9, right? You guys look like you're confused. Do we not remember that? Okay. So arachidonate is a 20 carbon fatty acid, 4 double bonds. It's derived from linoleate. I'm not a fatty acid person. The thing about biochemistry is there's somebody who studies this for their whole life. There's somebody who knows that molecule, like that's their existence, right? So they know it better than anybody else. They know all the enzymes and everything about it. That's what a PhD is, right? When you go to PhD, like
I studied prion protein. I studied one protein for 5 years, right? That's what I did. Years of my life, the production I can actually say is a paper, right? At the end of that time, I actually said, here's 2 years of my life. That's the thing about grad school, right? It's a good question. What is the point of that?
You know a lot about that.
I do know a lot about that. I do know a lot about topics, specific topics, but also we're adding to the body of knowledge. Every single one of us does a little bit, and by the end of the time, we have a lot. I think I've told you, we generate a textbook worth of information about chemistry every 2 or 3 days. All of this army of PhDs are out there working, slaving away. Amanda and I write 3 to 5 or 6. I don't know. Last year, Amanda wrote a lot of papers last year. That's the contribution that we do and we give. And certain people read them. We're ranked based on that, how much we produce, how much we publish. It goes into specific journals, right? So I don't know. I'm not a fatty acid person. I'll just tell you that right now. But this class is pretty broad, right? The idea is to introduce you to a bunch of topics. We've all been through satellite biochemistry. There's people out there who know all about these fatty acids, and arachidonate is a precursor for signaling molecules, the icosanoids. I'm saying that wrong, but they're signaling molecules and signaling hormones. We'll talk about those tomorrow when we talk about steroid synthesis. And they introduction of prostaglandins. Prostaglandins you may be familiar with. Those are some inflammatory cytokines. They're local hormones. They're short-lived. They affect nearby cells. And they're very, very important in signaling between cells. And they're very important in inflammation. So we'll talk all about that. Arachidonate is the precursor for all of these classes of molecules that we're going to talk about tomorrow. And that'll be part of the lecture that we talk about. So these molecules, and some of these are also important in other hormone cycles. So we got prostacyclin, prostaglandins, reboxanes, all derived from these prostaglandins. That all generates from this arachidonate. And we also can generate leukotrienes. You've probably heard of leukotrienes. They're very popular in the press. We're going to generate these from phospholipids and diacylglycerols. So that's the important part of arachidonate. It's a major precursor of the icosanoid hormones that include prostaglandins, which we'll talk about a lot tomorrow. Actually, maybe we'll talk about them today. Maybe more tomorrow, but we'll talk a little bit about them today. Get all my lectures blended together. They influence respiration, blood pressure, GI, inflammation, reproductive systems. A lot of your reproductive systems are associated and are triggered by prostaglandins. They're regulated by cyclic AMP. What is cyclic AMP? What is AMP? Adenosine monophosphate, right? So there's a cyclic version of that. That's a very important regulatory mechanism in a lot of systems. That regulates prostaglandins. Prostaglandins influence all of these systems, and they also play roles in the treatments and conditions for high blood pressure, ulcers, and asthma. Aspirin is actually one of the main regulatory entities in this prostaglandins. It alters the function of prostaglandins in your body. So here are some of them. We'll talk about how we make these tomorrow, those reactions, but here are the various. There's a prostacyclin, it's a vasodilator. Leukotriene B4, it's a pro-inflammatory signal. Is inflammation good or bad? Well, that's a good answer. What does it depend on? Okay, and what else? Where it's at? What else? What's the immune system really known for? What does it do? It fights infections. So it can be good and it can fight infection. It's also really known for overreacting, right? If you get a cut, sometimes it can just, you know. So how do we solve that? How do we normally treat that? You guys play sports? Ice, right? You cool it down, right? What else do you do?
Put it up.
Put it up, yeah, elevate it. What is that going to do? It's going to decrease the blood flow, right? Which will decrease the ability of things to get to it, right? Right? What does that do? Anti-inflammatory. It's going to affect this cycle right here. That's exactly what aspirin is, ibuprofen, all these things. They're anti-inflammatories. And so they're going to affect they're going to counteract some of these pro-inflammatory signals that you see that are prostaglandins. This prostaglandin E2 is something that induces labor. Right? So there's all kinds of, this thromboxane enhances platelet aggregation. So these things do quite a bit of localized signaling. And they're all generated from essential fatty acids, and we'll talk a lot about that. So aspirin, just as we mentioned, prevents the use of arachidonate as a substrate for the enzyme that generates the prostaglandin H2, which we talked about as an inflammatory agent. So, it can all block all of these COX-2 mechanism, peroxidases, all these other different mechanisms. It's an inhibitor of all these signaling mechanisms, and that can decrease inflammation along the way. So acetyl-CoA carboxylase is involved in all this. They're subject to regulation, right? That's the first step. So we're going to talk about the regulation of this pathway now. Carboxylase 1, it's inhibited by AMP, an activated AMP kinase, and it gets phosphorylated. So we can, once we phosphorylate this enzyme, it becomes inactive. That's generally how we regulate. It's the same slide. Must have copied that. Acetyl-CoA carboxylase. It's a dimer. It's 265 kilodaltons, which is an enormous protein. It has multiple subunits. Some of the cofactors are citrate. It actually can also create a filament out of this enzyme in the active form. And that filament then can generate huge amounts of the fatty acid and allow it to really become enzymatically active. It's only active in this form. So it's an inactive form, it's in dimers, and once we can make it into a filament, once we polymerize it, then it becomes enzymatically active. Citrate can partially activate this. This is another way that we can, even if it's phosphorylated, it can be partially activated with citrate. So if we have citrate in the, where is citrate found? So that can partially activate this, even if it's phosphorylated. It's another mechanism that it can be regulated. It can also be regulated by hormones, things like insulin, glucagon, epinephrine. Insulin stimulates the dephosphorylation and thus activation. Also can be stimulated if there's no fatty acids around. So these are some of the active polymer is activated by insulin, which removes that phosphate. Glucagon and epinephrine can also activate it, and we have an active polymer. Citrate can partially activate it. So those are the main regulatory mechanisms by which this whole process is regulated. Ethanol, which is your favorite drink probably, favorite beverage at times, it alters this pathway, leads to an excess production of NADH that'll inhibit gluconeogenesis, and you can get lactic acidosis. That's an excess NADH inhibits fatty acid degradation and stimulates fatty acid synthesis, which can lead to accumulation of fats in the liver, which is one of the reasons why you see liver disease in people who drink. So this process is affected directly through consumption of alcohol. Now, I don't think there's anything biochemically wrong with having occasional drinks. I think this is just more of an excess thing if you're doing it consistent. Liver can convert some of the acetate that's generated in this aldehyde dehydrogenase into acetyl-CoA, but it can't process the amount of NAD plus that's generated. That leads to ketone body secretions. That exacerbates the acidosis and lactate accumulations. That's kind of what gives you your problems. Acetaldehyde accumulates, very reactive, and causes loss of protein functions. That loss of protein function, if it accumulates, will cause your liver to fail, and that's probably some of the reason why you have problems. So, all right, that's the fun for today in